So a controlled genetic cross means to have control over really as much of this as you can. So as you've learned about scientific experiments, it's important to keep things constant unless you are deliberately trying to make them different. So with pea plants, you'd be controlling to have peas coming from the same species, being as genetically similar as each other as possible, except for the gene that you're trying to compare, to grow them under the same conditions and treat them the same way, and so on, so that you can isolate these genetic elements. So controlled genetic cross also has kind of a standard format here. So starting with a population of true breeding plants, shown here is, again, with this example of tall plants and dwarf plants, these would be true breeding individuals of those different strains or varieties of pea plant. And so starting the experiment means crossing plants that come from groups that have not been crossed for quite some time. And that is the parental or the pea generation, or maybe the pea generation, since you're working with peas. And two parental strains crossed with each other produce the first filial generation, the F1 generation. And this is one of those general principles that shows up in Mendelian genetics, is that the first generation offspring take on the phenotype of the dominant alleles. So they have two alleles, one from each parent, but they inherit this dominant allele that gives them the tall feature. And a subsequent generation, called the F2 generation, is made by crossing the F1 individuals by each other. So F1 by F1 becomes the F2. And another interesting pattern that Gregor Mendel observed was that the dwarf, this recessive phenotype, would show up again after being missing for a generation, would show up again. And not only that, but show up in a predictable frequency of 1 in 4 plants on average would have the dwarf recessive phenotype, and 3/4 would have the dominant phenotype. So let's say you had these tall pea plants, and we kind of determined that we could have two different genotypes giving us this tall phenotype, right? So what kind of genotypes could give us tall? They have to have at least one big T. That's basically it. So big T slash something. Sometimes you write that as big T slash blank, if you want to cover all possibilities. And then, of course, we have just the two alleles in this case. So big T, or big T, little t. So big T, homozygous dominant, or big T, little t, heterozygous. So again, looking at these plants, you don't know what their genotype is. How would you maybe be able to determine that? So one way to do this there we go, is to do a test cross. So a test cross has a specific definition. I think some people end up expecting it to be more of a general use term. Test cross specifically means to cross something, basically quite often with an unknown genotype, crossing that with somebody who is homozygous recessive. So a dwarf pea plant, in this case, we know that any plant that is dwarf has the homozygous recessive genotype. And for the rest of these, we don't know their genotype, but we know they have at least one big T. So they could be big T, or big T, little t. And based on the possible genotypes, we could work out the possible outcomes for this. So we'll kind of maybe get around to that again. Let's say we had this case where we had the dwarf individual as a known genotype, and let's say hypothetically the tall individual was heterozygous. So considering these adult plant, mature plant genotypes of being homozygous recessive and heterozygous respectively, then the diploid genotype for these plants produces the possible alleles that can end up in the gametes. So I put this slide together to illustrate that any one plant is producing lots of sperm cells and lots of egg cells. In this case, actually, one plant would produce both sperm and egg cells as a plant of this kind. And when we're looking at the possibility of combining two of these alleles into the next generation, we're kind of drawing from this set of possibles for one parent and this set of possibles for the other parent. So in the parent that's got the producing the egg cells in this cross, it's homozygous recessive. It's got only little t's to give. So we have a certainty here, 100% chance that this parent is going to contribute a little t allele to the offspring. This individual is heterozygous, has two kinds of t's, some big t's, some little t's. I've got 4 of each shown here, and those would end up in possible sperm cells contained within the pollen grains. And because we have 2 kinds of these, and a large number of pollen grains being produced, you can get predictive about what the chances are that any one of those pollen grain sperm cell alleles is going to get contributed to the zygote. So this ties back into what we saw before with the life cycle. Diploid mature individuals producing haploid sperm cells and egg cells, the gametes, and then those gametes combining together during fertilization to produce the next generation zygote. So this zygote would grow up into an embryo inside of a seed in this plant, go on to start the next generation. So what are our chances here? What are the odds that these 2 parents can produce a big T, little t offspring? So we see in this genotype, there's a little t, there's full chance that one of our parents is going to give little t's, and the big t's are half of the possible contributions from this parent. So we can predict that. We can actually predict this mathematically, and we can predict also the phenotype. So again, we're not in this cross, not actually observing T's floating around in anything. You couldn't put these under the microscope and see any differences in what their chromosomes look like or their DNA. The DNA is going to look the same, especially with the tools available in the middle of the 1800s. But we know based on deduction that they've got something different going on depending on whether they're big T or little t alleles. So the standard way to calculate the expected proportions of offspring is to put together a Punnett square. You've probably seen these before. And a Punnett square amounts to a square where you've got a number of possible gametes coming from one parent on one axis and the possible gametes coming from the other parent on the other axis. So a parent with a little t genotype is producing only little t alleles for the gametes. A parent with big T, little t genotype is producing either big T or little t alleles for the offspring, or for the gametes. All right, so what's shown here, I may not have the parents spelled out exactly. Plant on the left is contributing to what's shown there as parent 1 gametes. Individual on the right is contributing to the parent 2 gametes. And we're looking at one gene. Each gene has, each individual has 2 alleles. So we have 2 possible alleles here, 2 possible alleles here. Now a simpler way to do this And a method that I recommend that you do is that if you've got two of the same, you don't need to write them twice. So I think there's kind of a knee-jerk reaction, way that people put together their Punnett squares without thinking about them and say, okay, I got two alleles, I'm going to put them both on here. But if you put on two of the same, you're just writing the same thing twice. So the simpler way to do that is to write it once. So what this says is that this individual can produce only little t's, and we don't need more than one column here. Just one column will cover that. Every offspring is going to get a little t. And then on this axis, we have two possibles. So that gets us our possible combinations. So everybody gets a little t coming down and a big T or little t coming in from the side. The convention is to write the capital letters first, anytime that happens. And so this tells us the possible outcomes, and it also tells us that they're equally likely. So this is a 50% chance that each of those genotypes would be produced. And you could also write in your phenotypes, and we've ended up with a 50% chance that either of those phenotypes would be produced, right? So they're total independent genotypes as well as phenotypes in this Punnett square. So what I've shown you a couple slides back is what's called a monohybrid cross, kind of a standard investigation. We saw there with the P generation and the F1 generation. The monohybrid part here, I'm not always super fond of this terminology, but the mono part refers to one gene, and the hybrid part there is basically where you've got heterozygous individuals. So we did this before, but now we could write in their genotypes. We knew that the parental generation was true breeding, so we had homozygous dominant crossed by homozygous recessive. If you did your Punnett square for that one, that would be a 1-by-1 Punnett square, one kind of allele combined with one kind of allele. And so we know everybody in the F1 generation would be heterozygous, big T from the tall parent, little t from the dwarf parent. And the monohybrid cross gets interesting when you get to the F2 generation, where you can again tell the genotype for the dwarf plants, but we don't know exactly what's going on with any of these. We know that they were big T at least, and either big T or big T little t for these tall pea plants. But you can get in there and predict the fact that there would be 3/4 tall plants and 1/4 dwarf plants here, as shown here. So in this cross of the F1 by F1, then you'd be looking at, I guess I'll fill down first, then fill across, making sure my capital letters go first. So this would be the genotypes of the offspring. Of those, anybody who gets at least one big T gets to be tall. That's the outcome that we saw and would expect. And then one of these 1/4 would be dwarf. And then of the tall, I'm going to keep it with the fourths there. So out of everybody of the offspring, 1/4 of the offspring would be big T, big T. Just go ahead and stick with 2/4 of the offspring would be big T, little t. Both of those would fall into this 3/4 tall phenotypes category. And then 1/4 of the offspring would have the dwarf genotype. Again, you've probably seen that before, so hopefully that's pretty clear for you. Another way to look at crosses in Mendelian genetics is to get a little more complicated and do a dihybrid cross. So dihybrid cross, we saw monohybrid was one gene. A dihybrid cross involves looking at two genes at the same time. And so in this case, we've got two versions here of the peas. So now phenotypes of the seeds, we've got green and round and yellow and wrinkled. So what we know based on other information is that we have a gene affecting the color of the seed, green versus yellow, and another gene, round versus wrinkled. So using this information and what we've seen with alleles, we could deduce some information about our genotypes here. So gene notation, for some of our examples, you'll get to come up with that on your own. Knowing which one is the dominant allele is going to be important for this, writing the genotypes for your individuals. So let's isolate these one at a time. We've got green crossed with yellow and produced yellow, right? So what that tells us is that one of these plants, or one of these alleles, the yellow allele is dominant, right? So yellow, we're going to use Y for yellow because it's the dominant allele. And the green plants get little y. I put a little curly Q on some of my letters. They're, depending on your handwriting, sometimes you can't tell the difference easily between uppercase and lowercase. Do it for me, do it for yourself. Just a good habit to get into, making sure that you can tell your letters apart. So they look a little silly, but there's no mistaking that that's a lowercase y for me. Put a semicolon here to tell us that we've got a second gene. This is round versus wrinkled. We crossed the round by wrinkled and got round. So now we're going to do big R for round and little r for wrinkled. And that makes everybody in the F1 generation have one big Y allele from the yellow parent, one little y allele from the green parent, one big R allele from the round parent, one little r allele from the wrinkled parent. So that would be the genotype for both of those. Anybody in the F1 generation, there's two homozygous parents, and so there's only one way to get them crossed in their F1 genotypes. What's the F2 generation look like? Well, the Punnett square gets a little more complicated at that point. Clear our notation here. So this was the genotype we figured for the F1 generation. So they put their Y's and R's in different order than I did, but big Y, little y, big R, little r is the genotype for the F1 individuals. And how they produce their gametes is one of the R alleles and one of the Y alleles. So big R with big Y, little r with little y, big R with little y, little r with big Y. 4 possible gametes. And then the possibilities of them crossing become a Punnett square with a 4x4 layout. So big R, big Y, little r, little y, big R, little y, little r, big Y, and so on. So you can play that out. I won't do the whole thing because it's drawn on for me on the next slide. So this is already worked out for you. And they've done the genotypes and also the phenotypes. So dihybrid cross produces, in this case, 4x4 Punnett square gives us 16 outcomes to consider. And the phenotypes that go along with those. So anybody who gets a big R is a round seed. Anybody who gets a big Y is a yellow seed. And combination of little r's and little y's gets us wrinkled and green. And combination of big R and little y is smooth and green. Combination of little r and big Y is wrinkled and yellow. So we've got 4 different kinds of phenotype there. And what I want to wrap up with here is the independence of these inheritances. So the chance of producing a green seeded plant, if we just did that independently and we compared, let's say we're doing just the Y allele, we would predict a 1/4 chance of green and 3/4 yellow. And what do we find? We got 4 out of 16 here green. 4 out of 16 is also 1/4. And if you did the same thing for round versus wrinkled, you'd get the same prediction. So what we get with the Punnett square considering 2 genes at the same time is an output of how those phenotypes would mix with each other. And we'll get a chance to do some more of that. So combining these together, green and round, gets us something we could predict with the Punnett square, 3 out of 16. And then later we'll talk about how you can do that with a probability, more of a mathematical approach to that kind of question.